everyone for coming today. We're very lucky to have John Shurin with us, who battled lots of traffic to get here today. John did his undergraduate degree at McAllister College in biology and chemistry, and then he graduate work at University of Wisconsin and University of Chicago with Stanley Dodson and Matthew Leipold. Then did a postdoc at NC's not far from here, did faculty at University of British Columbia, and then now came to UCSD, so he's close by, except for the traffic.
Geographically.
Geographically. It was a great person to talk to. And we're lucky to have him because I think he has some of the most interesting ideas and papers on top-down versus bottom-up control and aquatic versus terrestrial systems and food webs and how diversity is regulated by consumer resource type relationships. And he's also one of the funniest people I know. So together it should make people talk.
Thanks, Van. Much appreciated. All right, well, it is great to be here, and it was an adventure. But yeah, I'm very excited to see old friends and meet and hear about what you guys are doing. I'm going to talk today about work that's been going on in my lab on the topic of the cumulative impacts of local and global environmental changes. And there's some really well-known sort of global changes like rising temperature variability, changes in precipitation, changes in atmospheric CO2. But there's a lot of changes that are taking place in the environment at sort of more constrained local scales that can be even more dramatic than many of these sort of big global things. And so a lot of the sort of unknown about what the ecological impact of environmental change is going to be deals with whether these sort of processes act to reinforce each other or interfere with each other, dampen each other, how these sort of different processes are going to play out when many things are going on at the same time. So I'm going to talk today about two projects, two main themes. One is asking how is diversity related to temporal variability in lake plankton communities, and in particular, how is diversity related to the pace and the kind of temporal variability, the rate at which the environment changes, whether it's on sort of short time scales or longer time scales, and how we describe that variability on different time scales. And then I'm going to talk about how multiple environmental changes taking place simultaneously in freshwater food webs interact with one another. And in particular, this is going to ask an experiment that asks the question of how does increases in temperature affect the expression of top-down and bottom-up control, the impacts of trophic cascades, and the effects of nutrients on pond food webs. So those are the two topics for today. Okay, so the first thing you need to know is something you already know, which is that all environments everywhere in the world are variable all the time. So throughout the day, over seasonal cycles, years, decades, millennia, everything changes. So all organisms are faced with an environment that is constantly shifting around them. And as ecologists, we're very interested in how this variability and how the fact that different environments are different in how variable they are. Some are more predictable, more steady. Some are more dynamic and more rapidly changing. How does that affect the structure and the function of ecological communities? So that's one reason we're interested in that question. The other is that the world is becoming a wilder and woollier place. Things are becoming more dramatic. The frequency of extreme events is going up. This is a plot for the US of the human population, the triangles, the number of weather-related catastrophes, sort of big biblical events like floods and storms and hurricanes and tornadoes and things like that, all the kind of stuff you hear about. And then the financial losses as a result of those events. So the frequency of extreme variation at least is going up. So at least on that sort of scale, the frequency with which these very, very extreme dramatic events take place is going up. Okay, so diversity is one aspect of communities that many of us are interested in. So the question then is how does environmental variability through time affect species diversity, ecological coexistence of species in communities? And there's two sort of categories of effects, and these are sort of make up the basis for the intermediate disturbance hypothesis, which is one sort of way of thinking about this. But one is that temporal variability can have a positive effect on diversity by preventing competitive exclusion through storage effects, temporal niche partitioning. So one sort of classic example is desert annual plants. Some of them like wet years, some of them like dry years. So if you have precipitation variation through between years, then no species can become a sort of competitive dominant. And this is just an example with two species of daphne in a lake where the environment is changing over time. One of them sort of declines and the other one sort of rises up and replaces it. So the idea is that if the environment were constant in this lake, one of these species might eventually come to exclude the other, and diversity would be lost. But variability can also, as you know, have negative effects on diversity by driving species extinct, exceeding their physiological tolerances, exposing them to periods of slow population abundance when they're vulnerable to stochastic extinction due to demographic or environmental stochasticity, reducing geometric mean fitness. And so the question that we're interested in is, what is the net effect of variability on diversity? Which of these two categories of effects is most important in determining the outcome or the relationship between diversity and variability in communities? So that's the question: how does environmental variability through time affect diversity? Before we can answer this, we need to consider two other things. One is what aspects of the environment, what kind of variability is important, and then second, on what time scale, because some things can show very high frequency, short time scale variation. Some things may show very long patterns of change over long periods of time. And so how do we partition variability among different time scales? So I'm going to describe a project looking at environmental variability in a series of lakes in the temperate zone of North America and Europe, looking at patterns of environmental variability with respect to diversity of the plankton communities that are found in those lakes. So first I'm going to describe the data set. So I compiled data from long-term monitoring programs of a whole bunch of lakes around the world. Some of these are sort of the classics, like the Wisconsin Lakes, LTR, the experimental lakes area in Canada, Lake Zurich and Geneva in Switzerland, a bunch of other lakes that have been very well studied for very long periods of time. And this is just sort of a standard thing that if you were a limnologist, you would go out and measure. This is the temperature depth profile. So this is depth in the water column. Sort of a typical stratified water column. You have a warm mixed upper layer and then a thermocline and then a bottom. Cold bottom temperatures. So from this we can extract the surface and the bottom temperature. We have similar, in all these data sets we have observations through time on, in addition to surface and bottom temperature, we have chlorophyll A, which is the abundance or biomass of algae, phytoplankton in the water column, total nitrogen, total phosphorus, two major nutrients that limit growth of plants, the vertical maximum and minimum of dissolved oxygen, conductivity, salinity, pH, and dissolved organic carbon, which comes mainly from decomposing terrestrial vegetation and has a bunch of different effects on aquatic ecosystems. And we also have observations of zooplankton species through time. So also measuring the abundance and diversity of the kinds and numbers of crustacean zooplankton, such as these ones. Oops, something's happening. Such as these guys here through time. So we have these observations from 53 lakes around the world. As I said, all in the temperate zone, mostly in North America, some of them in Europe. And these lakes were sampled between about 10 and about 40 years for a total of about 1,000 years of sampling across all of the lakes, or about 6,000, almost 6,000 monthly samples. So we took a monthly average to sort of standardize for the sampling interval. And then we asked, how does variability in all these things relate to the diversity of zooplankton that we observe? So we have to first figure out how to describe patterns of variability. And we did that using what's called a multiplicative model of variance decomposition. I'll describe it to you in an equation, and then I'll show you some graphs to show you what it means. But for every observation at every time step, so we have an observation at t, which is a measurement of something like pH. We calculate 4 components. We calculate the long-term mean, the average across the whole data set. We calculate the interannual component of diversity, which is the annual mean divided by the long-term mean. So if it's something like temperature, if it's a warm year, this ratio will be greater than 1. If it's a cold year, it'll be less than 1. We calculate the seasonal component, which is the average for that calendar month divided by the annual mean. So again, if it's June, it'll be greater than 1. If it's January, it'll be less than 1. And then we rearrange all this and solve for this E, which is this residual or unpredictable component. So that's the part of variation that's a part separate from any sort of long-term trends and separate from any sort of seasonal variation. So you can think of that as sort of short-term residual unpredictable variation. So we calculate all of those for every observation in every time series. And then the standard deviation across that time series is a measure of the variability on each time scale. So the good thing about this is it allows you to compare variability in things that are measured in different units, like temperature and pH. And so you're measuring in terms of a percentage of the mean. So each variation is a variation in terms of percentage of the mean. Okay, so what does that look like? So here's an example. This is a sort of a classic lake in Canada that was acidified by acid rain in the 50s and 60s and 70s. This was sort of the dominant environmental problem of that time. Emissions of sulfur and nitrogen from coal power plants mainly caused the pH to drop down to around 4 to 4.5. So a pH of 4 to 4.5 is extremely acidic. Very few things can live under those conditions. So no fish, no amphibians, virtually no insects, a few microbes and some bacteria and some algae, and that's about it. So this was sort of recognized as a problem and things were done about it, and over time the pH recovered. This is 35 years of data. So it increased over time, and here's the long-term mean here. So if we calculate the mean of each calendar year and divide it by the long-term mean, we can see that early on the pH was about 15 to 20% lower than the long-term mean. And later on it was about 20% above the long-term mean. And the standard deviation is 0.15, saying that the standard deviation between years is about 15% of the long-term mean. And you can appreciate that once we've taken out that interannual trend, there's not that much variation left over. So the variation around that interannual mean is not very large. So this is the seasonal component now. And there's virtually no seasonal variability, which tells you that there's not really high pH months and low pH months. There's variation within years, and that mostly falls to this residual or unpredictable component. So for this time series, the lion's share of the variation is interannual, as you would expect from the time series. There's a little bit of residual and basically no seasonal variation. So how does this let us compare different environments? So here's 35 years of pH in this lake that showed recovery from acidification. Here's another lake in Canada that was better buffered and never got acidified, so it has a stable pH over the entire period of observation. And if you look at the three components, as you predict, the lake on the left has much higher interannual variability than the lake on the right. The lake on the right actually has higher seasonal and residual variability, which you can sort of see because the line kind of wiggles around more on a shorter time scale than the one on the left. So what does this look like for things that show a more seasonal pattern, like temperature? So this is now surface and bottom water temperature, surface in green, bottom in blue. For ELA, Lake 239 in the experimental lakes area, it's a small lake in the middle of Canada in a very, in a continental climate. And in between, so we cut the data off between May and October because we had sparse observations in the winter. We did that for all of the variables. And you can see that between May and October, in the surface, it goes through about a 20-degree range of temperatures. You can also see that the green and the blue lines bump into each other, which indicates that the water column goes from being stratified to mixed during this period of observation as well. And this is Lake Washington, a very large lake in Seattle in a maritime climate, much more stable climate. And its surface water only goes through about a 10-degree range of temperature, so about half as much variation in temperature. You can also see that the surface of Lake Washington is slowly warming up. It's warmed by about 1.5 degrees or 2 degrees over the last 40 years or so, which means that the difference between the top and the bottom, which is the stability of stratification, has grown greater over time. And so if you look at the three variance components, the lake on the left is more variable on all three time scales. So this is interannual, seasonal, and residual for the surface in green and the bottom in blue. For the surface of both these lakes, the dominant signature of variation is seasonal, as you'd expect. If you want to know what temperature it is, the most important thing to know is what month of the year are you in. Not so much in California, but in other places very much. And this lake on the left, as you would expect on all three time scales, is more variable than the lake on the right. So the question then is, how does the diversity of plankton in these lakes, of zooplankton, relate to the long-term mean, to interannual variability, to seasonal variability, and to residual or unpredictable variability? So we use model selection procedures to identify variables that were predictive of the number of zooplankton species recorded in the lake. We use the number of species found on one day. We could use a number of species found in a year or over many years, and we get the same results. So I'll just show you for one day here. So I'm going to show you now the results of all these analyses. Okay, so I'm going to show you a bunch of graphs that look like this. So this is the number of zooplankton species going from about 4 to about 16 found in these lakes as the function of the average surface temperature. And there's a negative correlation between surface temperature and zooplankton richness. So lakes that have warmer surface water, on average, tend to have fewer species in them. And it's not really obvious why that should be, except that we also found that the lakes that had warmer surface waters, on average, tended to be less frequently mixed. They tend to be more consistently stratified. So they didn't go through periods of stratification and mixing as often. And those periods are really important. They sustain really different communities. They resuspend nutrients. They do a lot of things. They cause a lot of change in the communities. Next, there was a positive correlation between mean of pH and zooplankton richness. These lakes with low average pHs were acidified at one time or another, or they recovered from acidification. Sorry, each point is a lake, and the color indicates what data set it's from, because a lot of these lakes are sort of spatially aggregated. This is the mean across the whole, the long-term mean. So this is the mean of the whole time series. Yeah. Okay. So higher pH in general, you have more species. And then the other thing that was important was dissolved organic carbon. Now, DOC does a number of things in lakes. It fuels microbial production. It attenuates the ultraviolet radiation. It has a bunch of effects on zooplankton. So it's not really clear why you have more species where you have higher DOC, but that was a fairly consistent pattern. So we next looked at interannual variability. So this is now how much variation, this is the average variation among years. And there was a positive relationship between interannual variability and bottom temperature and zooplankton richness. Now, bottom and surface temperatures were well correlated with each other, so I don't think it really means anything whether we got bottom or surface temperature falling out as significant. But again, we found that lakes that were warmer on average had fewer species, and lakes that were more variable on average tended to have more species. So higher interannual variability tended to be associated with more species. Next, interannual variability in pH. So these lakes with high interannual variability in pH had few species. Those were, again, the ones that got acidified by acid rain and were recovered from such acidification, and they tended to not have many species in them. And next was a negative relationship with interannual variability in total phosphorus. Now, it's interesting, total phosphorus is an indicator of productivity or trophic state, so this tells you how green the water is. And we found no relationship between diversity and the average of productivity, but there was a negative relationship between diversity and the variability, the interannual variability in total phosphorus. So lakes like these that have high interannual variability generally underwent eutrophication or nutrient pollution, became excessively productive, or were recovering from such eutrophication, and they tended to have few species. So the average of total phosphorus didn't matter for diversity, but the variability in total phosphorus was important for how many species we found. Next was the seasonal variability. Again, seasonality and temperature. So lakes that have a large seasonal cycle and temperature tend to have more species in them and tend to be colder on average. Next was seasonality and pH. So again, a large seasonal variability in pH tended to be associated with fewer species. And finally, seasonality and DOC. So the long-term mean of temperature, pH, and DOC were important, and the variability. So lakes that were warmer on average had fewer species, more variable, more seasonally variable had more species, and the mean and the variability in pH and DOC also had sort of opposite correlations with diversity. And stop me if you have any questions. And finally, residual or short-term variability. This is short-term variability in bottom temperature. Again, a positive correlation between variability in temperature and diversity. And a negative correlation between short-term variability and total phosphorus. Now, this short-term variability in total phosphorus is sort of indicative of spikes of production associated with runoff of nutrients from the watershed. So you get a big sort of bloom of algae. So you have very sort of spiky, highly variable algae abundance. And in lakes like that, you tend to have very low diversity. You tend to have dominance by a small number of species. So the conclusions then from this section is that in general, temperature variability is good for diversity. So lakes that are more variable in temperature tend to support more species, whereas water chemistry variability tends generally to be bad for diversity. Lakes that are more variable in water chemistry tend to have fewer species in them. And there's a few reasons why we suspect this might be the case. One is that temperature variability is more predominantly seasonal. So if you have a more pronounced seasonal pattern, then it's more predictable. Organisms can evolve to time their life cycles to correspond to the appropriate thermal conditions in which they are able to live. The other thing is that the range of temperature variability, even in lakes like Lake Washington that have shown steady increases in temperature, those increases are really small compared to the overall variability in temperature. So an increase of 1 or 2 degrees is small when your annual range is much bigger than that. Whereas the water chemistry variability is much, much more dramatic. So going from a pH of 7 to a pH of 4 is a really, really dramatic change. And you're far outside the natural range of conditions that most of these species ever would have encountered during their evolutionary history. So there's very few species that have evolved in any sort of environment that has a pH that low. The other thing which I didn't show you, but we found as well, is that environmental variability was often as or more important than average conditions for predicting diversity. So the amount of variability that an environment experienced could be equally as important or more important. And so if we want to understand sort of how diversity is going to change in future environments, we need to understand how regimes of variability are going to change. Mike?
John, are there any differences in the, I guess, the taxonomic membership across these lakes, and are there any phylogenetic effects that relate to these diversity patterns?
For sure. I mean, a lot of this is driven by things at high DOC. Yeah, so there's definitely a phylogenetic signal. There's copepods that do really well at high DOC. So it seems like different foraging strategies work in different environments differently depending on, so if you're a really high DOC lake is really brown tea-stained water, has really low light penetration, and so you can have a bigger body size, you're less apparent to predators. So there are definitely patterns in composition, and those are definitely related. So related to dominance of cladocerans versus copepods, which are sort of the two main. That's the main. If you made the phylogeny of all these zooplankton, that would be the deepest split, is between copepods and cladocerans. They're all crustaceans, but yeah, There's definitely a phylogenetic signal to this. Yes. Oh. Hmm?
What about the geographic signal?
Oh.
You mentioned continental versus maritime climates.
Mhm.
There are a lot of the difference in temperature variability. So if you just take, for example, all the highly continental lakes, you have the same effect.
Well, yeah, I mean, so we coded these by color, and that's a great point because you can see that a lot of the differences are about differences between regions. So like all these pink lakes are in the same region of Canada, right? So it's not like, so some of these patterns are consistent within regions. Like these yellow lakes are the Wisconsin lakes, you can kind of see there's a negative correlation here, maybe if you squint just right. But a lot of these differences are about, these lakes are all together, these blue lakes, and these pink lakes are all together. So this overall pattern is driven by, in many cases, by variation between regions, not within regions. So there's a lot more variation in the kind of conditions you get across these big continental regions than there is within any. All the lakes of one region will be really pretty similar to each other compared to the continental variation. So that's a really important point.
I have a similar question with the plot on the left. It seems like the green dots are driving that trend, whereas everything
Yeah.
kind of at the beginning is fairly spread out and doesn't really have.
Right. Yeah, I mean, it's always a question with these kind of things, how deep you want to delve into trying. I mean, yeah, so it seems like all of that, yeah, if you took, these are the experimental lakes area, if you took those out, you would have no pattern there. That's certainly true, and there's a number of these patterns that are true like that. I mean, that's why we haven't, the broad brush conclusion is about temperature versus chemical variability. We haven't tried to sort of dive too deeply into the sort of particulars about what explains each individual pattern, because even though we have 1,000 years of sampling from 50-some lakes, it's not really that many data points, right? Like if we had similar data for thousands of lakes, we could get into what's really driving each one of these patterns. But with this is the most data there is anywhere in the world, but even so, it's not that much for teasing apart all the sort of particulars like that. All right. Okay. All right. So that ends our first section. Now I'll be talking about some experimental work asking how multiple environmental changes taking place simultaneously interact to alter the effects of top-down and bottom-up control in pond ecosystems. So why do we focus on this? Well, there's three sort of main environmental changes that we know are taking place at sort of a global level. One is that we know that the weather is getting, on average, warmer, and it's getting wilder, it's getting more variable through time. This is a very general pattern that's taking place everywhere, so we're interested in how temperature is going to affect these kinds of species interactions. The others, we know that in both terrestrial and aquatic ecosystems, top predator communities are being really dramatically altered. And this is very sort of obvious in the ocean. This is Daniel Pauly's sort of classic depiction of fishing down marine food webs, where through industrial fishing has sort of serially depleted the biggest fish like sharks and tunas here at the top that are longest lived and most susceptible to fishing. and then over time sort of shifted towards these smaller and smaller fish at lower and lower trophic positions. This is trophic position in the food web here. And what a lot of us are really interested in is what's going to happen down here in the microbes and the communities, the organisms that are really responsible for sort of global chemical and energy cycles. So the things like phytoplankton and bacteria that are really participate in global cycles much more than other things. The other sort of global change is that humans have liberated nutrients to fertilize our farm fields. And in so doing, we've introduced a lot of the chemicals that limit primary production into the environment and elevated their availabilities in general. So in post-industrial times, the amount of carbon circulating in the biosphere has gone up by 13%. Nitrogen has doubled, increased by 100%, and phosphorus has increased 400%. And this is just sort of one sort of example of the effects that's having. This is nitrogen fertilizer application in the watershed of the Mississippi River. Those put it up, so that's fertilization of things like soybeans and corn. Lots of those plants are really bad at taking up those nutrients. Lots of them wind up running off through all these rivers and eventually come out into the Gulf of Mexico here in New Orleans and fuel this giant algae bloom here. And this is the dead zone of the Gulf of Mexico, where you have big surface blooms of phytoplankton. Those phytoplankton die and sink to the bottom and fuel anoxia as they decompose as bacteria break them down. So the deeper waters here become anoxic, and that's driven off things like fish and killed off things like invertebrates. So all three of these things are happening simultaneously all around the world. And why do we expect that changes in nutrients, predators, and temperature should interact with one another or not? Well, there's some reasons to expect that they would. One is that different organisms of different sizes and therefore at different positions in food webs vary in their sensitivity to temperature. So this is from data from Mary O'Connor showing the resting metabolic rate, the respiration rate of zooplankton as a function of temperature going up along the x-axis here, and then the photosynthetic rate of phytoplankton, of algae that those zooplankton are eating. So both of these things increase with temperature. You get higher respiration rate and high photosynthesis rate, but they do so at different rates. So zooplankton respiration is more sensitive to temperature than is phytoplankton production. So they increase more rapidly. So you might expect that the top-down effect of zooplankton grazing would be higher out here at higher temperature, where their metabolic rate is greater. Alternatively, these zooplankton might run out of food quicker if they're not able to feed fast enough to satisfy their metabolic demands here at higher temperature, if their food production is not keeping up with their metabolic needs. So we might expect to see stronger top-down control by consumers at higher temperatures. But we know that your metabolic rate doesn't go up sort of indefinitely with temperature, that everything reaches some sort of thermal maximum and then starts to decline. And different species may have different thermal maxima. It's possible that bigger things like fish may have lower thermal maxima than smaller things like phytoplankton or zooplankton, in which case we might expect to see stronger top-down control with increasing temperature in this region. And then eventually we may get to some temperature where we start to lose these higher trophic levels faster than these lower trophic levels. So there's a number of possibilities as to what might happen to top-down control. And similarly to bottom-up control, the role of nutrients in controlling productivity at different trophic levels may be differently sensitive to temperature. Okay, so to ask this question, my students and postdocs and I did an experiment in these large sort of artificial pond mesocosms. These are 1,000 liters, they're 2 meters across. And they're big enough to sustain a sort of diverse community of many species of zooplankton, phytoplankton, benthic bugs, snails, tadpoles, self-sustaining populations of little fish like these sticklebacks. And we factorially manipulated temperature with this very safe method of all these extension cords with large aquarium heaters dangling in them. So each one has an aquarium heater. And that warmed them up by about 2.5 to 3 degrees. And they followed the sort of daily and annual cycle, but at about 3 degrees warmer than the ambient tanks. And we manipulated the presence of these ferocious three-spined stickleback predators. These are predators of zooplankton and benthic bugs as well. And we also crossed that with the presence of nutrients. So we had 2 by 2 by 2, three factorial treatments: nutrients, temperature, and fish predators. And we sampled over 17 months weekly to look at how the effects changed seasonally as things moved on. So I'm going to take you through the results of this. All this work was done by a team of super postdocs who are now all gainfully employed professors, so I'm happy about that. So Jesse Clausen, Pavel Kretina, and Hamish Grieg, who are all in my lab at the time. Okay, so I'm going to show you the results for algae abundance on one date, and then I'm going to show you across the whole experiment. So this is now a plot of the chlorophyll concentration, so the biomass of algae in the water, in the tanks without fish in white and with fish in red. So the difference between the red bar and the white bar is the sort of strength of top-down control, the impact of fish on phytoplankton at the base of the food web. And this is in the control where it's at ambient temperature and there are no nutrients added. And so this is on in the middle of February up there in British Columbia, so it's cold but not freezing. But most of these zooplankton have gone dormant or disappeared. So the abundance of herbivores is very low. And as a result, there's no difference between the tanks with fish and without fish. So there's no apparent trophic cascade in sort of ambient conditions. If we add nutrients and fertilize, now we see a different effect where we do see a difference between fish and no fish, and that's because when we fertilize, we increase primary production, and where the zooplankton are the hot top trophic level, they graze down that algae and they keep the standing stock of phytoplankton at about the same level as in the control, but where fish are present, they're controlling zooplankton, and therefore that excess production builds up in the algae. So this is an example of an interaction between nutrients and fish. So the effects of fish are stronger when we've added nutrients than when we haven't added nutrients. This is at ambient temperature then. What happens when we warm things up? When we warm things up, we see a very different kind of effect. We're now, in the absence of nutrients, but at higher temperature, we do see a trophic cascade. We see an effect of fish on algae abundance. And again, we see that effect become bigger when we fertilize and add nutrients. So basically, this is an interaction between temperature and fish in that higher temperatures magnify the effect of fish on algae abundance. And that's because at higher temperatures, the zooplankton persisted through the winter better. So they maintained their abundance. They didn't decline in the cold season as much as they did where we weren't warming them up. And if you look at this just in terms of the effect of temperature on phytoplankton abundance, what you can see is that now these arrows connect the same treatments in the cold temperature to the ambient temperature to the warm temperature for each treatment combination. The red ones are with fish and the blue ones are without fish. And you can see that the effect of warming depended qualitatively on whether fish were present. So when fish were present, warming increased algae abundance. We had more algae present when fish were present. When fish were present, and where fish were absent, warming decreased algae abundance. We had lower biomass of algae when we warmed things up. So the effects of temperature depended qualitatively on the nature of the predator community that was present in these tanks. Okay, well that's on just one date, and we sampled every week. So what happened? So now I'm going to take you through the effects over time. And what this shows is what we did. So we used a sort of proper mixed effects model to test the significance, but then to look at the effect size, we did an ANOVA on each sampling date and looked at the parameter value as a measure of the effect size of the treatment on that dependent variable. So now a positive parameter value means that we increased algae abundance in that treatment, and a negative one means that we decreased it. And these are the sort of the confidence intervals, so this gives you an idea of where those effects would have been significant had we only sampled once, which we didn't. So this is the effect of fish in blue, nutrients in green, and warming in black. So you can see early on, the nutrient effect sort of appeared and persisted through the rest of the experiment. So we fertilized, took a little while for the algae to take off, then they took off, and so we had higher algae where we fertilized. The fish effect appeared a little sooner and then was consistent throughout the rest of the experiment. In the first summer, when things were relatively unproductive, there was not a big warming effect. There was no significant warming effect, really. And then there was no effect throughout the winter. And then as it got warm in the second summer, the net effect became negative. So we started to see lower algae abundance in the tanks at high temperature than at low temperature. So warming, on average, across all the treatments, decreased algae abundance, but only during the second summer there. So that's one way we can look at the effects over time. We can look at the interactions in the same way to ask how these treatments are either magnifying or dampening each other's effects. So we can look at the parameter estimates for the interaction terms. So now this is the interaction between fish and nutrients. And you can see that it was mostly not significant, and then in the winter it became significantly positive and stayed sort of significantly positive. So that's the effect I showed you, stronger trophic cascades when we fertilize. This is now the fish by temperature effect. So you can see that that effect was not significant in the first summer, and then when it got cold in the winter, it became significantly positive. This is the data that I showed you where warming magnified the effects of fish. And then as it warmed up in the spring, it dropped out again and became not significant. And this is the interaction between warming and nutrients. You can see that it was mostly not significant in the first summer throughout the winter, and then in the second summer, it became significantly negative. So if you're thinking of this in terms of does warming stabilize or destabilize food webs, you can see that it's destabilizing in that it magnifies the effects of predators. So it increases the importance of top-down control, but it's stabilizing in that it dampens, it reduces the effects of nutrient fertilization. So the effects of nutrients are less pronounced at high temperature, but these effects are really seasonally dependent. So the stronger top-down control occurred only in the winter. The stronger bottom-up control occurred only in the summer. Okay, so those are one way of looking at the stability of production. The other is to look at temporal variability over time in primary production. So we looked at the annual fluctuations in the abundance of algae. Again, we're talking about chlorophyll. And so this now shows for each line here is one replicate pond, the concentration of chlorophyll in the control, the fish, the nutrients, and the nutrients plus fish. And this is all at ambient temperature. And what you can see here is the effect of what's called the paradox of enrichment. That when we fertilize with nutrients, the abundance of algae becomes much more variable over time. So algae abundance starts to fluctuate wildly. Here in these unfertilized tanks, they're all relatively consistent within a treatment and within a tank. When we fertilize, we get these big blooms and crashes up and down, and these are asynchronous. So one tank will be really green, another one will be really clear within the same treatment, and that's sort of a signature of intrinsic instability or sort of cycles or chaotic dynamics driven by fertilization. So that's sort of the classic expectation from the paradox of enrichment, that fertilizing should make the biomass of producers more variable over time. Now, what would you expect? What would you expect warming to do? Would you expect it to magnify variability or reduce variability? Magnify? That's what I would have expected, and that's what most people would expect. And anytime someone asks you a question like that, you know that you're about to be proven wrong. And so that's not what we found. So these are those same treatments where at low temperature we saw these really wild dynamics of algae, and at warm temperatures we see that those disappear. So we don't see those big algae blooms and busts the same we did at low temperatures. And so warming in many senses may be stabilizing depending on how it affects the algae and the things they interact with. There's a number of reasons that we might expect warming to be stabilizing. If at high temperatures, for instance, the zooplankton metabolic rate increases more than its feeding rate, then the zooplankton are not able to feed enough to keep up with their metabolic needs. And so their ability to sort of zoom up towards high population density and cause crashes in the algae may be reduced. Their population growth may be reduced. But the result of this is that warming and nutrients interacted, and warming reduced the sort of paradox of enrichment type effect on instability of primary producers. Okay, I just have a few more, and this is just showing those same results. This is the standard deviation over time in chlorophyll and the mean of chlorophyll. These are for the warm tanks, and these are for the cool tanks. So you can see at the same sort of level of productivity, the same algae abundance, at low temperature you have a much higher variability than you do at high temperature. And this is just sort of what those extremes look like. So this is sort of a very unproductive tank. You can see here's our heater on the bottom, our little periphyte and growth tiles, various instruments. And this is a super, super, super productive tank where if you stuck your hand in the water, you wouldn't be able to see it anymore once it was a centimeter or two underneath the surface. Okay, so I'm going to describe a couple other things that we measured in this experiment. One was the effects of the treatment on movement of matter between aquatic systems and terrestrial systems. So aquatic systems are linked to terrestrial systems both in that they receive terrestrial detritus that comes into lakes and ponds and rivers and is either mineralized, decomposed, broken down, releasing CO2 to the atmosphere, or else builds up in the sediments where it can be stored for a very long time. One of the sort of main frightening positive feedbacks of climate change is that higher temperatures can accelerate decomposition. So a lot of the organic matter that's currently locked up in things like peat bogs in Russia and Canada and places like that may start to decompose and be released out into the atmosphere. Lakes and ponds also release a lot of material in the form of the bodies of organisms that have complex life cycles, where they lay their eggs in aquatic systems, grow up as juveniles and larvae, and eventually emerge as adults, taking all that material that they've acquired with them. So we measured both of those things. We measured emergence of insects and frogs. We put in frog egg masses and measured how many adult frogs we got out. And we had these little hats for catching bugs as they came out, so things like damselflies and chironomids and things like that. So I'll just show you the results for emergence. We also measured effects on decomposition, which I will not tell you about, but I'd be happy to talk about if you want. So this is the effects on emergence of insects and amphibians, frogs, specific tree frogs. In the top here, so this is for ambient temperature and warm temperature with nutrients in black, without nutrients in white, and no fish and with fish. So you can see there's a positive effect of warming on bug emergence. So we get more bugs out when we warm things up. There's a positive effect of nutrients. So we generally get more bugs when we fertilize. And there's a huge negative effect of fish. So the fish eat up all the baby bugs, all the larvae, and prevent emergence of the adults. So they prevent the adults escaping. And there's a fish by nutrient interaction, in that we see a big effect of nutrients when fish are not present, but when fish are present, they eat up all those extra bugs. So that's the effects on bug emergence. For frogs, the main effect was really about fish, is that frogs and fish do not get along. We know this from many examples, that when we had fish present, that we got virtually no frogs emerging. And when fish were absent, the frogs did just fine. Okay, so the final thing I'm going to tell you about then is the effects of the treatments on the shape of the rest of the food web in these ponds. And this is sort of the classic depiction of a lake food web from Lindemann, 1942, the sort of inventor of food webs in a way. And this is sort of his depiction of how organisms in lakes interact. You have a pelagic food chain here, which is what most people think of as the food chain, where you have phytoplankton at the bottom. They're eaten by zooplankton, they're eaten by bigger plankton predators, and they're eaten by these big swimming predators, by which he means big fish. You also have a benthic food chain, where you have pond weeds or vascular plants, or things like periphyton, things that live in the benthos attached to the bottom. They're eaten by browsers like snails or tadpoles or insect larvae. They're eaten by their own predators. And then these big swimming predators swim around and feed in both of these environments. But all these things die, and they contribute to the ooze, which many of you have probably encountered in your own experience with lakes, which is the dead organic matter sitting at the bottom, which is being mineralized by bacteria. And the sort of great insight of Lindemann was that he recognized that these bacteria are sort of recycling dissolved nutrients, which are then a sustaining production of these primary producers in both of these food webs. So sort of recognizing the central role of bacteria in aquatic food webs was Lindemann's sort of main contribution. And there's one other group of organisms that Lindemann didn't know about or didn't appreciate their role, and that is the viruses. So we have the microbial loop, but we also have something called the viral shunt. And you can think of viruses as sort of competing with grazers for phytoplankton. So a lot of viruses attack either bacteria or phytoplankton. They lyse phytoplankton cells and release dissolved organic matter, which can fuel heterotrophic bacteria. Those can also be attacked by viruses. And so basically, viruses are preventing, in a sense, the export of organic matter to the deep part of the ocean, to the sediments, either in the ocean or in lakes and ponds, by lysing these cells and releasing this particular this dissolved organic matter, and preventing flow of nutrients up to the higher trophic levels in the food chain there. So we measured abundance of all of these groups, and this is sort of a mind-bending graph, but this is how we depicted the effects of the treatments on all of these different groups of organisms at the same time. So this is not a spider web, but it is a 6-axis graph, and so the value along each of these axes tells you the biomass of each of these groups in the presence of fish in red and the absence of fish in green. And where we have, so we've drawn boxes around these. Where there's a red box, it means there's a significant negative effect over the whole experiment. And where there's a green box, there's a significant positive effect. So what we found is that fish make the food web bottom heavy. So they decrease the abundance of zooplankton and benthos, which are fish food. That's what fish eat. That's what you'd expect them to do. We see increases in phytoplankton. So this is the trophic cascade. Phytoplankton bacteria, and periphyton. That's all completely expected. What's not expected is we found that fish also decreased the abundance of viruses, which is very surprising because most viral ecologists would tell you that the viruses should do whatever the bacteria do, that most of the viruses are eating bacteria, and we would expect their abundance to track the bacteria. So this suggests that the fish have some kind of effect on the viral lytic cycle through the composition of the community of bacteria or algae or something that we don't know about, and we would have to be much more clever with molecular techniques to find out what it was exactly. But in any case, that's the pattern that we saw. Warming had sort of the opposite effect of concentrating biomass in the upper trophic levels, not so much by increasing the abundance of consumers as by decreasing the abundance of producers. So both the phytoplankton and the periphyton were significantly negatively affected by warming. There was no significant effect overall throughout the whole experiment on zooplankton, benthos, or bacteria, and there was a significant positive effect on the abundance of viruses. There were more viruses at high temperature, but they decreased the abundance of both primary producer groups, both pelagic and benthic. Primary producers were less abundant at higher temperature. And finally, fertilization made all of these different groups go up. So we had greater abundance of everything in the food web. The things like unicellular things tend to increase more than the consumers, the benthic bugs and the zooplankton. But in general, everything became more abundant. Now this is interesting because all three of these treatments, fish, fertilization, and warming, increased net ecosystem production. So the amplitude of the daily oxygen cycle was higher in all three of those treatments. But they did so in really different ways because they really affected different organisms at different positions in these food webs very differently. So they all had very distinct effects on the shape of the food web, but they had a very consistent effect among all three of them on the total ecosystem respiration, the total amount of energy processed by that ecosystem through photosynthesis and through respiration. So the conclusion from this experiment then is that we saw stronger top-down control, stronger trophic cascades at higher temperatures in ponds. So warming amplified trophic cascades, but only during the cold time of the year, not during the warm time of the year. We saw weaker effects of eutrophication, smaller effects of nutrients, but only during the summer, during the hot time of the year. We saw stabilizing effects of warming on eutrophication. We saw less sort of violent, dramatic phytoplankton blooms and crashes at higher temperature rather than at lower temperature, which was surprising. One of the effects of warming often is an increase in outbreaks of things like cyanobacteria, and we did not see that. We found that fish are really sort of the border guards of lakes and ponds. They prevent biomass exchange with terrestrial ecosystems. They prevent emergence of both insects and amphibians. And they also prevent decomposition of detritus. So they actually, by eating all the bugs, they prevent detritus from getting broken down because those bugs are what chews up that detritus. And we found really distinct effects of these three different sort of environmental changes on the shape of the food web, on the distribution of biomass among the different components of these food webs. So this has sort of been the inspiration for what we're doing now in my lab. So we're looking at, so these are sort of abstracted experimental systems. These effects are, even in a 2-year experiment like this, these effects are sort of short-term effects. So we've been asking about the effects of temperature on the expression of top-down control by comparing all of these same different groups of organisms and ecosystem rates, things like pelagic production and respiration, CO2 exchange with the atmosphere, sediment accumulation, abundance of all these groups of organisms in lakes in Yosemite and in the Sierras with and without fish at different elevations. So this is my PhD student Celia Simmons' work. So she's been out sampling lakes in Yosemite. Here she is measuring periphyton growth. And these lakes, so all these lakes are historically fishless. They were all stocked with fish for recreational fisheries, sort of indiscriminately. Some of them were not stocked, some of them have had the fish removed, so some of them are now fishless. So Celia has sampled lakes with and without fish across an elevational gradient, which is of course a natural temperature gradient. So this is just an example. So she sampled between about 2,000 and 3,300 meters of elevation. The fish lakes are in blue, the non-fish lakes are in red. This is mean temperature. This is just showing that it gets colder as you go up the mountain. We figured that we got that one right. And then this is decomposition here. So this is decomposition of leaf detritus. So we put in packs of leaves like these in mesh bags, in these lakes and came back and weighed them after a month. This is one that's decomposed a lot. This is one that's decomposed a little. And you would expect decomposition to increase with temperature. And instead we found mostly in negative, we found that decomposition was highest at the top of the mountain at low temperature, and mainly in lakes without fish. So these lakes at the top of the mountain that are really cold, that don't have fish, have just tremendous abundance of benthic bugs. The bottom is just covered with chironomids and caddisflies and all kinds of insects. And much more so than the lakes with fish. The fish eat all those bugs. And we found, interestingly, that decomposition was faster at low temperature and in the absence of fish. And there was interaction between. So in the fish lakes, there was basically no change with temperature. In lakes without fish, there was a decline in decomposition. So this suggests that shifts in the communities along these sort of natural environmental gradients give rise to outcomes that are very different from what you'd expect based on sort of short-term experimental manipulations. That changes in the composition of these communities, these lakes that have been separated, that have been sitting there at different temperatures for very long periods of time and have assembled very different communities. We see that the ecosystem effects and the changes in the food web and the ecosystem may be very different from what we would have anticipated based on our experiment. But this is sort of testing the idea that the expression of trophic cascade changes with elevation. Yes?
Is the oxygenation condition at the bottom different?
That's a good question. Many of them are quite shallow. So they're generally, the ones that I can think of off the top of my head had very little difference between the top and the bottom. They have very little organic matter. They're very, very, very oligotrophic. And so they generally, many of them are above tree line, and so they have very little decomposition in them. So those ones have generally the same oxygen top to bottom. The lower elevation ones may have, because they have more DOC in them, but I can't tell you right offhand whether that's true or not. But yeah, I'll get back to you. Okay, so why is all this important? Well, it's important because these changes in ecosystems may alter the capacity of ecosystems to serve as sources or sinks of carbon to the atmosphere. So the carbon that CO2 we release into the atmosphere, only about half of it stays in the atmosphere. The other half is taken up by both terrestrial and aquatic ecosystems. And changes in nutrient availabilities, predator communities may affect the productivities of these ecosystems and their capacities to serve as sinks or sources of carbon to the atmosphere. So that's why we're very interested in how things that affect ecosystem configurations interact with climate to determine their sort of role in the global carbon cycle. Okay, so just give you the general conclusions that climate variability affects diversity. In general, temperature fluctuations are good for diversity or are associated with higher diversity. Water chemistry changes are bad or associated with lower diversity. And temperature interacts with top-down and bottom-up control in ponds. In that warmer temperatures strengthen top-down control and generally dampen bottom-up control. So we see a shift to weaker bottom-up control and stronger top-down control at higher temperatures. Okay, so these are my collaborators on this work and the people who gave me money back when I was in Canada, and thanks for having me. Priyanka.
So you have this sort of at high temperature or like warming causes a stronger top down, and then with cool or ambient it's stronger bottom up. Is that between seasons when you see that?
That's a good question. So we saw that as an effect. So the warming effect was
But it was continuous though, right?
Yeah, that was continuous. It was a 3-degree warming that was 3 degrees in the winter and 3 degrees in the summer.
Okay, so extrapolating from that to say a seasonal environment.
Well, this is a very seasonal environment. So let's see. We should be able to tell that from looking at this. Let me see. Where are you? Blah 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 blah. Okay. So this is the effect of warming and nutrients. And that's sort of the effect. So the green line is the effect of fertilization on algae, and the blue line is the bottom up, and the blue line is the effect of fish. And they don't seem to really change that much seasonally. I mean, at the beginning they took a little while to get going. We didn't want to just sort of blast them with huge amounts of nutrients. So they took a while to get going, and then you can kind of see that there might be seasonal patterns in here, but
the fact is you had a 3-degree increase the whole time, so I wonder whether the increase in the winter had a fertilization off.
Yeah.
And then the summer all might do it, right?
Yeah.
Sometimes those things make a difference.
Absolutely.
Either a warmer winter or a cooler summer. Or
Yeah, I mean, you expect the exponential loss to be nonlinear. I mean, going from 20 degrees to 23 degrees is very different from going to 10 degrees to 13 degrees, for sure. And yeah, I mean, for the trophic cascade, I mean, we saw that the effect was definitely strongest when it was cold. And I'm sure that that's generally a 3-degree change. Yeah, when you're in the middle of summer and it's 25 degrees every day, that effect is not going to be very strong. I'm not sure if
no,
you can.
I'm interested in sort of whether you would see that in a sort of a typical seasonal environment under.
If you just
the-
if you just measure, yeah, I mean,
They cancel out, or you'd have one being strong during, say, the colder period of the year, and the other sort of integrated across the year.
yeah.
But it's harder to try. It's a bit hard to extrapolate from this.
Yeah, I'm trying to think how to extract it from this data. I mean, that's what these main effects, I mean, this is like the difference between fish and no fish, averaged over all the other treatments, right? So that, so I would expect the fish effect to be strongest in summer, in general, because there's not that many grazers around in winter. So based on that sort of ad hoc argument, I would expect to see that. I'm not sure, yeah, I don't think we have any way of seeing that in these results the way we plotted them. But maybe if we were cleverer, we could figure something out. Yes?
I'm a fish biologist, so I'm very interested in all these fish interactions. But Kirsty, I missed the very beginning of your talk. Did you say something about how you decided how many fish, for example, ought to be in your artificial ecosystems, compared with other things, and what levels of nutrients?
Right.
So, I'm just wondering how much other variability there would be depending on the starting conditions.
Sure.
And then with respect to the current project, which the graduate students working on up in Sierra, the fish that are stocked in those lakes are mostly trout.
Yeah, all
And
trout.
they're not, that's what Cal Fishing Game does.
Right.
They're certain, and they're not sticklebacks.
No.
Or anything like that.
Yeah.
And so you've got, apples and oranges.
Yeah.
And so, how important are those kinds of interactions?
Oh, hugely. So the answer to the first part. Okay, I'll go back to the beginning. So the sticklebacks, all the ponds we set them up, we just took a big bucket of mud from a real pond and dropped it in there and let whatever hatched out. For the bugs, so the bugs just came and laid their eggs, and we had as many as 20 or 30 species of insects that just came and laid their eggs on their own. And then a whole bunch of algae and zooplankton hatched out of the sediment that we put in there. The fish we put 5 adult sticklebacks in as a sort of, which is low. I mean, there are places in BC where you can take a net and go like that, and you'll get 30 sticklebacks, right? In a thing the size of, your bathtub. So that was a fairly low abundance of sticklebacks, but we knew from other work that they would have the effect that they had. There was some interesting stuff that happened with the sticklebacks, which is mainly that they're predatory on their own babies, so they're cannibalistic, and so they generally didn't reproduce, except at the high temperature in the water, in the fertilized tanks, where the adults all got some parasites and all died after they reproduced. So their babies all did great. So then we had really high abundance of babies because their parents died of some horrible disease. So unlike humans, they don't seem to like having their parents around. So that was really interesting. In terms of the Sierras Lake, so yeah, we're discriminating between fish and fishlets, and definitely among the fish lakes, some of them, there's 20 guys fishing every day, and they seem to be pulling trout after trout after trout out of them. Some of them, it's hard to tell there's any fish there at all, but people assure us that they are. So we can see in the, you can see it in the zooplankton whether there's fish or not. There's certain species that will not be there if there's fish present. So, and things like frogs as well. It's also interesting, you can tell whether there's fish by the birds around the lake. So there's rosy finches that feed on emerging bugs. And if it's a fishless lake, they're all around the shore feeding on the bugs that come out. And if there's fish in the lake, there's none of them because there's no bugs coming out for the reasons that we found in our experiment. So you can kind of diagnose whether it has fish or not by some, without actually catching any fish, which is good for us. Yes.
Can you please go back to the slide and show a minute ago when you were analyzing
Priyanga's. This main effects or the interactions?
It was the interaction?
Okay, here's,
Yes.
there's all the interactions.
So the warming time fish one, especially, has this kind of hump that it goes up and it goes
Yeah.
down. And because it can go on, and that's just no way to tell whether is this a dynamical transient or is this driven by seasonality. But do you have any thoughts, like, is this just is this a seasonal effect or is it a
Yeah, I'm pretty sure it's a seasonal effect. I mean, so dynamic. I'm not sure you mean by dynamical. If that's.
I mean
like,
that that often a system will respond one way in short term and a very different way.
Right. So if we had no, if it was consistently at that temperature, would it be? Yeah, I don't think we have any way really of knowing that. I think it's seasonal in the sense that the seasonality of the zooplankton was really different at high temperature. And so at low temperature, in the middle of the winter, there's just no zooplankton around. Like there's nothing there. And if you warm things up, there's a bunch of things that stick around that do better at higher temperatures. So I think it's seasonal in that sense. Whether that means, what would happen in a sort of different season, these are like Arctic ponds where, in the middle of summer, it's the same temperature as this. I don't know. I mean, there's no way of knowing. Does that answer your question? Kind of. I don't know if I can do better. But yes?
If some of the lakes are above tree line,
Yeah.
the detritus you use exists in all those habitats?
Right.
All of the detritus the same across?
Right, so we used the same detritus. So we got willow leaves, which are across the whole elevation. So that's why we chose it, because they are everywhere. But, I mean, so one thing that's definitely true is that at low elevation, the amount of detritus is much, 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 much greater. The high elevation lakes have virtually no detritus in them. There's almost no vegetation. The shoreline is like lichens and sedges and nothing bigger than that. So they get very, very, very little detritus. So whether, so that was part of why it was so surprising that there were so many detritivores apparently in those lakes, or when we put detritus in, things ate it like crazy. So I don't know what those are doing normally when we don't, I mean, there's some detritus, but very, very little in most of those places. So I don't know. I mean, that's a mystery that I don't know the answer to, why those lakes had such high, I mean, they have huge, if you look at the bottom, it's just crawling with bugs. There's tons and tons and tons of bugs. And I can't tell you why, but that is what it is.
And even within that, though, that we would expect that, as temperature increases, we would see that tree line creeping
Yeah.
as well, so that may be an
Yeah.
interesting effect in and of itself.
Absolutely. And I think, the main confounding factor with our temperature gradient is DOC, is that the lower elevation ones have much higher DOC than the high elevation ones. That's the main confounding factor with temperature. All right, thank you.